Good morning. It's a true pleasure to introduce Elizabeth Phelps. I have known Liz since some of the earliest moments of my career, starting when I was a graduate student at Yale. At the time, she was an assistant professor at Yale, and she was already known as a pioneer in the study of emotion and its brain bases in humans. Liz was one of the mere handful of scientists in the 90s to start studying the neural mechanisms of emotion in people as compared to in animal models. And she used a brilliant combination of theories and methods drawn from experimental psychology and behavioral neuroscience. And her work has taken her down a path, including continuing in affective neuroscience, producing some of the early work in social neuroscience and neuroeconomics. And it goes without saying, all of you know Liz. I'm not going to talk about all of her accomplishments, but she has been the president of Social and Affective Neuroscience Society, the president of the Society for Neuroeconomics, and the president for the Association for Psychological Science. Her work, simply put, is insightful, elegant, and convincing, and it always has been. On a personal note, I've had the pleasure of knowing Liz as a dear friend as well, and she is as supportive and loyal a mentor as anyone I know. She does her best to support the careers of budding scientists. And perhaps most importantly, she is hands down one of the most generous human beings I know.
Oh. Well, thank you, Lila. That was so heartwarming. And I also just want to take this opportunity to thank Michael and the organizers for this conference. It's just been a kick-ass conference. I've loved it. Thank you so much. And I'm honored to be here today. So I'm going to talk to you today about our research looking at the mechanisms of threat control. And I just want to start by kind of giving you a little bit of my evolution in the world of emotion and memory. And it was really my studies of memory as a graduate student that inspired my interest in emotion as a postdoc, particularly the observation that when we think about our lives, the things that stick out to us are, the emotional things. And this led me to think about studying emotion, but I really wanted to do so based strongly in a neurobiological model, even though I was studying humans. So when I got into this domain, one of the things I did was I tried to look for one of the best possible neurobiological models in non-humans that I could draw on for my investigations in human research. And this has led me to work with Joe Ledoux, who I've now collaborated with for 25 years, and you will see his work interspersed in and out of my talk today. So when I started in this discipline, I started looking for research in non-human animals where I had a good neurobiological model to build on, but my goal was really to translate this to humans. And so really when we started out, we tried to do tasks that were similar to what they could do in a rodent model, but in humans, and by humans here I mean undergraduates. And but ultimately, of course, we're not necessarily interested in how much humans look like rodents. We're really interested in how do these systems come into play in the complex social and emotional lives that are typical of human experience. So I've tried to take this trajectory from animal models to similar systems in humans to situations that may be unique to humans. And I think you'll see that thread throughout the talk today. And the topic that I'm going to focus on today is learning about threats, both how to acquire threats a little bit. I'll just review that work very briefly. That was my earlier work. And I'll focus mostly today on how do we now control learned threats, because this is perhaps the more important question because it has clinical relevance. But learning about threats is very important, how we learn about them, how we change how we think about threats, for a few very obvious reasons. One, the things that are dangerous to our survival are extremely important to know about, right? If threats tell us things that might hurt us. And two, sort of maladaptive threats in the inappropriate context, threat reacting and the reactions in the inappropriate context is sort of the basis for a lot of emotional disorders. So if we can understand how we can control threats that are no longer appropriate, things we've learned to be threatful that no longer are, we may be able to think of new interventions for those types of disorders. So the basic model that I'm building on is a model of threat learning, Pavlovian threat learning. Here's a slide I borrowed from Joe Ledoux years ago. I'm going to mirror Dan Schachter and go over here. Here is just sort of Pavlovian threat conditioning. You're all familiar with this. You play a tone to a rat. They may react, but no strong reaction. You pair that tone with a shock. After a few pairings, the tone itself comes to elicit a range of defensive responses, conditioned responses consistent with fear and threat reactions such as freezing, increases in blood pressure, heart rate, respiration, stress hormone release. Our very first studies tried to do similar things in undergraduates. Take an undergraduate, pair a neutral stimulus like a blue square with a mild shock to the wrist. After a few pairings, we see a range of responses consistent with a threat reaction. The primary one we measure, and the one I'll talk about today exclusively, is changes in the skin conductance response, an indication of autonomic nervous system arousal. And doing these types of works, we were able to verify what we knew from animal models by Joe Ledoux, Bruce Kapp, Mike Davis, and others, that the amygdala plays a central role in the acquisition, storage, and expression of this type of simple threat association. But then, when we thought about people, we thought about what about things like this? So I don't know if anybody in the room knows what this is. I don't know what this is. I actually took this off of a website that teaches children about germs. And germs are an interesting thing. So you have to teach children that germs are bad, that germs can harm them, that germs are threats. We don't recognize these when we see them because we don't see them. You might recognize this one. Anybody know what that is? Ebola, right? We saw that all the time in the Ebola epidemic. But we don't recognize these things because we have no perceptual experience of them, yet some people become so afraid of germs that they will wash their hands until they're raw. And this just points out how flexible threat learning is in people, in ways it's not in other species. That our imaginations, things we can be told about, things websites teach children about, can create threats that can be useful, but can be also very maladaptive. So one of the first questions we wanted to ask is, does this simple model of threat learning that we know quite a lot about from animal models apply to cognitive and social means of threat learning? So we could do something like this. I could simply say, here you are. I pair a blue square with a mild shock, and now you show a physiological response to the blue square by itself. Or I could say, when you see a blue square, you might get a shock. And if I do this, the physiological response I observe is virtually identical, with small exceptions to what I observe if I actually pair the blue square with the shock. Or I could say, watch this guy. What happened to him is going to happen to you next. All right. That took a long time to make a point. Sorry about that. But what we know from this, right, is that the amygdala plays a critical role in these types of threats as well. So when it comes to instructional threats, it's only the left amygdala that seems to mediate the physiological expression, perhaps because that's linguistically encoded. But observational threats are also, you don't actually need to have a bad experience. Observational threats also robustly engage the amygdala. So this phylogenetically older mechanism of Pavlovian threat conditioning that we can study across a range of species seems to play out in humans in ways that make it much more flexible for us. We seem the social communication of threat, the linguistic communication of threat also relies on this system. And that's good and bad, right? It's good that we don't actually have to be hurt to know the threats in the environment. It's bad because things we can imagine that never actually could possibly occur can create threat reactions in us, and that may lead us to things like anxiety disorders. So I want to, over the last decade or so of my research in this area, we focused on not how you acquire threats, but actually how you now diminish them when they're no longer adaptive, when you've now learned that this thing that you once thought was dangerous is now safe. And So now I'm going to focus for the most of this talk on how we now think about the mechanisms of threat control and what are some of the novel techniques we're looking at to more persistently and effectively inhibit maladaptive threats. So there's two techniques for threat control that are currently used in the clinic. One is extinction, and this has formed the basis for exposure therapy. When the stimuli that used to predict a threat no longer predicts a threat, you essentially take away the unconditioned stimulus. And the other is cognitive emotion regulation, when you learn to think about something differently to change your emotional response. So we started out this line of research by looking at the neural mechanisms in humans of these two types of techniques for controlling threat reactions and looked at the similarity between the two. And so in extinction, here I have my cartoon subject. We have a blue square paired with shock during acquisition. And then immediately after this acquisition period, we stop giving the shock. That's our first extinction trial. We bring participants back a day later, and we have a second session of extinction. And this is the skin conductance response. I'll present this a few different ways. Here I've plotted the stimulus paired with shock, in the CS plus in the dark bar, and the stimulus never paired with shock, our control stimulus in CS minus in the lighter bar. And what you can see is we see robust acquisition of this conditioned threat reaction. We see some extinction the first day. And here we see a return of the threat response. And so this is a very typical response. We know in extinction learning that essentially what happens is it's not that you've now rewritten the original memory. You've now created a second memory that the stimulus is now safe. And sometimes with the passage of time, changes in context, we can get recovery of the threat reaction. We see that robustly here, and then continued extinction. We know from animal models quite a bit about the neurocircuitry of extinction learning. So again, this was the basis for examining the brain changes in humans. And I'll just highlight a few aspects of this. So we know, primarily from the work of Mike Davis, that the amygdala plays a role in the acquisition of extinction. But importantly, that the infralimbic cortex, and here I'm going to call it the ventral medial prefrontal cortex in humans, is necessary for both the learning and the retrieval or recall of this extinction learning. So when we looked in humans, what we saw in the amygdala here I've plotted in red the bold response during acquisition, and here during extinction we see a slight decrease in amygdala activation during extinction, and this predicted early extinction learning. And here in the ventral medial prefrontal cortex, we see greater activity to the CS minus and the CS plus in early extinction, and this diminishes as extinction progresses. So in other words, we see an increase in bold signal to the CS plus during extinction learning. So this roughly is consistent with what we'd expect from the animal models. Essentially, in the animal models, if you stuck an electrode into the infralimbic cortex, you'd see an increase in firing in the infralimbic cortex during extinction and the recall of extinction. But we wanted to move on to now a situation that was more typical of humans, or unique to humans, I should say, which is being able to now use your thoughts to change how you react to something. And so I put this up here because it's sort of similar to this phrase, the glass is half full, the glass is half empty. So essentially, you can change your emotional response to something by reevaluating it from associating it with negative affect to neutral or positive emotional terms. And the way that we did this in this sort of simple threat learning paradigm that we used was on half of the trials, when stimuli saw one stimulus, a blue square paired with shock, and a yellow square never paired with shock, they saw the word attend just before they saw this stimulus. And prior to the scanning session, they were told, when you see the word attend, I want you to try to focus on your natural feelings when you see these stimuli. But if you see the word regulate, I want you to try to use the color of the stimulus to think of an image of something that's calming, something from nature. So we didn't tell them what the image should be. If it was a blue square, it could be a beautiful water scene, a yellow square, a field of daisies. But ahead of time, they practiced coming up with this image when they saw the word regulate. And what we saw, and here I'm plotting again, the stimulus never paired with shock, and the CS minus and stimulus paired with shock separately. What we see is individuals are able to diminish their physiological response to the stimulus paired with shock by using this regulation strategy. When we looked in the brain, in the amygdala, here I've plotted what we saw during extinction. So again, an increase in bold signal when you're acquiring a conditioned response, a decrease in bold signal when you're extinguishing the conditioned response. In emotional regulation, when you're attending to the emotional significance of the stimulus, we get an increase in bold signal. When you're regulating your response to that stimulus, we get a decrease in bold signal. And when we looked in the prefrontal cortex, we saw two brain regions that showed greater activity when you're regulating your emotional responses than when you're attending. One is the dorsolateral prefrontal cortex. We did not see this in our extinction study. This is a region that's been hypothesized in other studies of emotion regulation to play a role in the sort of online reinterpretation, the cognitive control that it's needed to think about this thing differently. And the ventral medial prefrontal cortex, the same region that we saw in our extinction study. And when we looked in the region of interest from our extinction study in the bold pattern for extinction and emotion regulation, what we saw was here during acquisition, we see this decrease to the CS plus that diminishes during extinction. And it's also there when you attend to the emotional significance of the CS plus, and it diminishes when you now regulate your response to that stimulus. So this, and when we did connectivity analysis using that seed, what we saw emerging was the dorsolateral prefrontal cortex and the amygdala. So this suggested to us a network in which we know the amygdala plays a role in the acquisition, expression, and storage of these simple threat associations. The ventral medial prefrontal cortex plays a role in inhibiting the amygdala to allow the expression of extinction learning. And this same circuitry seems to be used when you now use your thoughts to inhibit your threat reaction to the conditioned stimulus. So essentially, emotion regulation, we think, is co-opting this phylogenetically older circuitry of extinction to reduce your emotional reaction by using your cognitions. And I should say this was work by Mauricio Delgado when he was a postdoc in my lab. So we know that fear and threat responses following extinction and emotion regulation are susceptible to relapse. We call this fear recovery, and this is following the passage of time, that spontaneous recovery like we showed in our first extinction study, contextual shifts, we call that renewal or re-exposure to the unconditioned stimulus. And another potential mechanism by which we can have relapse is under stress. So I've highlighted how the prefrontal cortex plays a role, a critical role, in the both extinction and emotion regulation of no longer adaptive threat reactions. So we wanted to ask the question, does stress impair the inhibition of threat responses? And here I've borrowed a slide from a review by Amy Arnstein. And we know that stress has a number of impacts on the brain, and it can vary depending on the magnitude of the stress. But one of the most robust findings of stress is that even relatively mild stress can impair prefrontal cortex function. So we asked this question, does stress impair the inhibition of threat responses for extinction and emotion regulation? I'm just going to show you our emotion regulation studies today. So in this particular study, we changed our conditioned stimulus now to two pictures of snakes, one of which was paired with shock, one of which was never paired with shock. That was our acquisition. We then did what was called a cognitive restructuring technique session. We actually worked with a clinical psychologist who does cognitive therapy to come up with a way to do something a little bit more realistic in terms of this technique as it relates to cognitive therapy. And the way this worked is the participant was asked to give their subjective ratings of the emotional intensity, the range of emotions they felt when they saw these stimuli and what was the intensity of that emotion. We then gave them a session where we talked about the relationships between the thoughts and emotions and how sometimes when you think about things differently, it can actually change your emotional responses. And we said, okay, tomorrow you're coming back into the laboratory. You're going to do the exact same study you did today, this one here. I want you to come up with ways you could think about it differently to change your emotional response. So they might come up with, well, the probability of shock was really low. It was actually 33% on the trials that were paired with shock. It's only a picture, the shock is separate, there's no uncertainty about what's happening tomorrow. Whatever they felt they could use the following day. Then participants came in and they got put in one of two groups. For one group, we induced a stress response. We did this by sticking their hand in freezing water for 3 minutes. I'm going to give you everything we can do to scare you in a laboratory, except this is not the best stress induction technique in humans. The best one is to have people give a talk and look disapproving. Just saying. But that leads to like social rejection and worry and what is it called? Thinking about it too much anyway. So I'm not going to do that. So we stuck their hand in freezing water, and then the other group stuck their hand in room temperature water for 3 minutes. We then did another conditioning session and measured cortisol throughout. So here I've plotted for you on the left the difference in cortisol pre-post, the pre-post session. So here we see in the no stress group, even though we did threat conditioning, and we know that leads to a physiological response, sympathetic arousal, it doesn't lead to HPA axis activation. We didn't see any increase in cortisol. But we saw a robust increase in cortisol in the stress group. Here is the efficacy, sort of the proportion decrease in the conditioned response from day 1 to day 2. And the no stress group was able to use these strategies to decrease their conditioned response, whereas the stress group was not, even though they felt like they were doing so. And interestingly, this is also extended to subjective reports of how they felt about the conditioned stimuli. So in other words, they rated the intensity less so if they were in the no stress group than if they were in the stress group. So this suggests to us that stress impairs emotion regulation. I didn't show you this work, but also it impairs extinction and recall, so you show greater spontaneous recovery when you're stressed. And we think this is perhaps by altering prefrontal cortex function. And this was work by Candice Rayo, who's now a postdoc, and Candice will be on the job market next year, and she is awesome, just letting you know, if you happen to see her application. So, as I mentioned, following extinction and emotion regulations, the two techniques that are now currently used in the clinic, threat reactions are susceptible to relapse under stress and in a number of other circumstances. And it's thought to be that this is reflected clinically in the relapse rate following exposure and/or cognitive therapy. Now, these techniques are effective, they are useful, but they have too high a relapse rate and they do not work for everybody. And this has prompted me and a number of other people in the field to say, what can we do better? So I want to talk about that for the rest of this talk. Essentially, how can we enhance the persistent control of threat using novel techniques? And I'm going to discuss 3 different techniques we've used in the laboratory. The first is trying to target reconsolidation, which you've already heard about a little bit today during this meeting from Lynn Nadell in the beginning and then others during some symposium. So I'm not going to review this in detail because you've heard quite a bit about it, but the notion of reconsolidation is that every time you retrieve a memory, now that memory once again becomes fragile and susceptible to change, as opposed to sort of memories being stored once and stable over time. And interest in this topic was renewed when Kareem Nader, who was a postdoc with Joe Ledoux, did the following study. So he took rodents, he paired a tone with a foot shock. The rodents then, he waited a day for that threat memory to consolidate. He brought rodents back into the laboratory. He played the tone, and then he injected into the lateral nucleus of the amygdala a protein synthesis inhibitor that was supposed to block the reconsolidation of that threat memory, the restorage of that threat memory. And he brought the rodents back a day later. He played the tone, and it's as if they never had any experience of the tone paired with shock. So this was very exciting to us. We were collaborating with Joe then as well, and so we really wanted to translate this to humans. Of course, the problem is you can't do this in humans. We can't inject these protein synthesis inhibitors into amygdala. So we had to find another technique. And the other technique that we finally settled on after trying multiple things was the one that Lynn Nadel talked about, is that really, in one way, we think of reconsolidation as a system that allows for the dynamic nature of memory. It allows us to update memories at the time of retrieval with information in the environment that may be relevant for their future use. And so we thought, can we update memory with this sort of threat memory with new information about the value of the stimulus? And this was inspired by work by Marie Monfils, who's in the back right there. Hi, Marie. And so this is what we did. So participants came into the laboratory. They saw two stimuli, three stimuli, two of which were paired with shock, one which was our baseline, never paired with shock. I called this one the CS plus R for the reconsolidation CS plus, this one the CS plus extinction for the E for this extinction CS plus. We waited a day for these threat memories to consolidate. We brought participants back. We then gave one single presentation of this reconsolidation CS plus. This was essentially thought to trigger the reconsolidation process. Then participants underwent safety training. This is essentially identical to extinction training, but the difference between the CS plus R, CS plus E, is that we think the reconsolidation CS plus is undergoing safety training during the restorage of this memory during the reconsolidation process. Whereas this stimulus here is undergoing standard extinction training, which we know results in a second memory trace that then competes with the first for expression. Participants came back a day later. They were given some shocks to reinstate the conditioned response. We then presented the stimuli again and looked to see, did we see recovery of the threat response for these stimuli? So here are the results. In orange is the stimulus that underwent safety training during reconsolidation. In black, the stimulus that underwent safety training, underwent sort of standard extinction training, and in gray, the stimulus that never was paired with shock. This is the end of acquisition. I should say this looks really pretty because we only selected people that actually acquired equivalently to the two CSs. And so we did not, because we thought as a precondition to looking at recovery. And then this is the end of extinction. They extinguished the conditional response to all stimuli. And here's the first recovery trial. What you can see is robust recovery to the stimulus that underwent standard extinction training. No evidence of recovery for the stimulus that underwent extinction during reconsolidation. And we also then had another extinction session. This is the end of that session. I put this here just to let you know that in another version of this study, we actually brought participants back a year later, once again, did a reinstatement of the conditioned response and showed once again that we saw reinstatement for the stimuli that underwent, in the group that underwent standard extinction training and not the group that underwent extinction training during reconsolidation. When we looked at the brain for brain regions that differentiated responses to these two stimuli, again, and this is during extinction, the one that underwent safety training during reconsolidation and the one that underwent standard extinction training, the only region that emerged was the ventral medial prefrontal cortex. And if I look at the pattern of response in this region from late to early extinction, what we see is this increase in bold signal, much like we would expect from our other studies. So in other words, as you're extinguishing, you get an increase in bold signal in this region if it underwent standard extinction, but not if it underwent extinction during reconsolidation. So this, is consistent with the model. And again, we can't prove this is the case without having more neurobiology in non-human animals, but it's consistent with the model. Essentially what we're doing is not inhibiting the threat reaction to the stimulus that underwent threat conditioning during safety training during reconsolidation, but actually sort of rewriting, in a way, the memory so that it's no longer threatful. And just to confirm, we did a PPI analysis, and we saw differential ventral medial prefrontal cortex between the amygdala, between the amygdala and ventral medial prefrontal cortex, which was stronger during extinction than during reconsolidation. And this was work by Daniela Schiller, Jonathan Kanan, and again inspired by Marie Monfils, who's in the back. So this was very exciting to us. We've now replicated this study 4 times in my lab, 3 of which have been published. And we thought this is a great technique to think about translating to the clinic. But this initial study was published in 2010, and it's now 2018. And as of yet, there's very limited evidence that targeting reconsolidation is successful in clinical symptoms of fear-related disorders. So there have been a couple large reviews on this. There have been some exceptions. So there's a study by Zhu at Peking University who looked at using this type of technique with a reminder and then doing exposure therapy to reduce craving in drug addicts. A recent study from Barbara Rothbaum's lab showed that you can find some success in patients with a phobia of heights, but it only extends to reducing the physiological response, not the clinical symptoms of phobias. And using another technique, propranolol, which we have not had success with, there's been some suggestion that you can have some success with other types of phobias, but fairly limited success and also often not extending to clinical symptoms. And when we look at this work, our particular studies and other studies as well have sometimes been replicated, sometimes not replicated. You can do something as simple as asking people to cognitively rate whether they expect the shock. It can change the efficacy of this paradigm. There are a number of boundary conditions that have been identified in animal models that can limit reconsolidation success. The age of the memory, the strength of the memory, and again, just different labs are finding different things. One thing that might play a role in this, we think, is generalization. So when we do reconsolidation, when we target a memory via reconsolidation, I'm asking you now to retrieve a specific memory. One thing we know about anxiety disorders is that they tend to generalize memories more than other folks. Let me give you an example of this. If you had a bad response to this dog, would you then be afraid of this dog? The answer is of course not, but some people have a fear of dogs altogether from one bad experience. They've generalized now their fear from a specific instance now to a whole class of stimuli. And when you study this in the laboratory, you do tasks like this. You take a tone, say a 1,000 hertz tone, and you condition an animal or a human to pair this tone with a shock, and then you test other tones. And here's a non-generalized threat response, and here's a more generalized threat response. And patients with anxiety disorders tend to show more generalized threat responses. In addition, there's a number of other circumstances that can lead to generalized threat responses. One of them is whether you have a high-intensity US versus a weaker US. So in humans, we are, by definition, required to use weak USs, right? We are ethical scientists, right? We can't do really, really scary things. But we did our best. So we did a study where we gave two shocks. So there are two groups. One got what we called a low-intensity unconditioned stimulus, so it was a moderately unpleasant shock that they got. And participants always set the level themselves to tell us kind of how unpleasant it is. Or they had a shock that was highly unpleasant, and then we combined that shock with a loud aversive noise and a picture of a snake coming at you. That's like the best we could do. That's about it. So again, we had two stimuli. So one, a 1,000 hertz tone paired with shock, and then a 550 hertz tone never paired with shock, our control stimulus. So here's what we found so here's the conditioned response. So this is the skin conductance response to the stimulus paired with shock, the 1,000 hertz tone versus the stimulus never paired with shock. And we saw roughly equivalent acquisition of the conditioned response, but when we did a test of generalization, we played all these tones in between the CS minus and CS plus, and what we see was in the high intensity group, we get much more generalization of the threat response. So if it's a very aversive event, we're going to have a generalized threat response. And then the question becomes, does targeting reconsolidation alter generalized threat memories in the way it does for specific threat memories like just a blue square? And this we investigated with another technique of looking at generalized threat memories. Here participants saw categories of stimuli, birds and fishes. So is Marijn here? Marijn Kroos? So Marijn's in the back. He did this study. Marijn's Dutch. We were in the lab. He said the word fishes. I said, Marijn, it's fish. Trying to correct his English. And then we looked it up. Actually, it's fishes. If it's multiple species, it's fishes. I never knew that. Okay. Anyways. So we have birds and fishes. And one of them was paired with shock. The other was never paired with shock. Then one of these categories, we took the prototypical exemplar in birds, it's a robin, of that category to reactivate the conditioned threat memory. And then they saw a movie, which they always do in the 10-minute interval that we have, and then they underwent extinction for both categories. On the third day, they came back, some shocks to reinstate the threat memory, and again, we measured for recovery of threat responses. So the filled-in bars are the stimuli that underwent had the reactivation cue, the CS plus and CS minus, that was that group. The non-filled-in bars are the extinction group. What you can see here, I'm just plotting early and late acquisition, the differences between CS plus and CS minus. We see robust acquisition of the conditioned response in both groups. Here's the reactivation cue. And now here what we see is we have extinction in both groups. Here's early and late extinction. Here's the end of extinction. We get extinction of the conditioned response in both groups. And here's our recovery test. It's identical in the two groups. So for this generalized threat memory, doing the reactivation of the memory and then extinction during the reactivation period did not result in what we think is sort of a rewriting of the threat memory, perhaps because it's not a very specific memory. So as I mentioned, there's several boundary conditions that identified in animal models and humans that limit reconsolidation success. One of them may be that the increased threat generalization with traumatic events may make it difficult to target specific threat memories with reactivation. And this has now led to sort of what we're doing now, which is trying to think about in reconsolidation, how do we overcome these boundary conditions, but also all there other alternative techniques we should think about to control threat responses that may be a little bit more robust to different circumstances. So I want to talk about two additional techniques at the end of my talk today. One of them is stressor controllability, and the last we call novelty-facilitated extinction. So stressor controllability is really inspired by a sort of old literature on learned helplessness. You probably all know what learned helplessness is. It's when you would take an animal, you would shock them, and it sort of uncontrollably, they couldn't do anything about it. After a while, the animal stops to respond. It sort of became a model of depression. And we know that sort of uncontrollable shocks, a lot of stress leads to depression-like, anxiety-like symptoms in other species. But there was a flip side to this literature that people are less familiar with. So relative to an animal that never received shocks, animals that received shocks but could control when they got them actually showed better performance. This led to sort of some resilience. So controllable shocks actually led to a better performance than receiving no shocks at all. And so we know with stressor controllability that previous history of stressor controllability may facilitate future threat control, and of course the opposite, learned helplessness, lack of controllability, may diminish future threat control. And this has also been shown in studies of avoidance learning in rodents. So we want to ask the question, does avoidance modulate threat recovery in humans? So the way we did this, participants came into the laboratory, again, one stimulus paired with shock, another never paired with shock. During what I'm calling the threat regulation session, one group of participants had now the opportunity to, when they saw the conditioned stimulus, to engage in an action that would let them avoid receiving the shock. So the participants had to learn through trial and error that they had a cursor here, and if they moved this dot over here, then they did not receive a shock. So that was the avoidance group. There was also a yoked, what we called extinction group. They had to sort of follow the dots around, but none of their actions actually let them avoid the shock. And they were yoked to the avoidance group so that they got the similar number, so each participant got the similar number of shocks as one of the participants in the avoidance group, and all participants learned this response. So the end of this session was essentially extinction learning. Participants came back a day later. We then looked at threat recovery, so again, seeing all the stimuli without a shock to see, did we see a return of the conditioned response? So here, I'm plotting here now the differential response to the stimulus paired with shock, and then one never paired with shock, so this is a conditioned response. For the avoidance group and the extinction group, both groups acquired the conditioned response. This is the end of the regulation period, so both groups were able to diminish the conditioned response either through avoidance training or through extinction. But only the extinction group showed evidence of recovery of the conditioned response. So one of the things we know about the literature on stressor controllability from animal models is that it generalizes broadly to subsequent behavior. So not just sort of avoiding the one stimulus that you're afraid of, right, but to subsequent behavior broadly. So a group that has no avoidance shows greater freezing than a no stress group and greater social defeat, but a group that has avoidance actually shows less freezing and less social defeat. The group that had previous avoidance shows better future avoidance learning, more social exploration than a group with no stress, which again is also better than a group that had no avoidance. So we wanted to ask the question, do experiences of control in humans modulate subsequent threat expression that happens later on as well? So here we had participants come into the laboratory. They were given a grid like this, and they were told you can use the arrow keys to explore different actions. And they would receive a shock every 2 seconds when this grid was on, unless they learned to shuttle to the other side of this box. And they had a number of trials like this. So that was our escapable group. We also had a yoked inescapable group that, again, was told to use, to explore different actions, but their actions never had anything to do with the presentation of shocks. They got the same number of shocks as a participant in the escapable group, and we had a control group that never underwent this manipulation. A week later, we brought them back to the lab, put them in another room, and did a standard threat conditioning procedure. So again, one stimulus paired with shock, another stimulus never paired with shock, an extinction session, and 24 hours later, a measure of threat recovery. So this is what we found here. I'm plotting the conditioned response, a stimulus paired with shock minus a stimulus not paired with shock. So we see acquisition in all three groups. Here's the end of extinction training. And here, no one here has the ability to avoid anything. This is the end of extinction training. So we see extinction in all three groups. It's somewhat better in the escapable group. And here's the test of extinction recall the next day, the first few trials. What we see is we see robust evidence of spontaneous recovery in the group that underwent inescapable shocks, some recovery in the control group, and no evidence of recovery in the escapable group. So we looked to see what factors might modulate the ability to both learn avoidance and show success, this resilience with avoidance. And one of the things we saw was that people who both were able to avoid and felt like they were able to avoid the shocks the first day showed lower threat expression a week later. People that couldn't avoid the shock but felt that they could avoid the shock actually showed no benefit. Right? It was just whether you had control and you felt like you had control. And the other thing we found is that sort of this trait-like internal locus of control, how much do you think you have control in general in the world, predicts how well you do on avoidance learning tasks. So in terms of looking at the neurocircuitry, we know a little bit about this from the animal work, particularly Justin Mascarella, who's here, I think, somewhere over there. He was a postdoc with Joe Ledoux. He was our collaborator on these projects. And so we know from some early work from Joe's lab that when you engage in an avoidance response, it engages the pathway between the lateral nucleus and the basal nucleus of the amygdala up to this striatum. That damage to this pathway impairs avoidance, whereas the standard threat reactions require the central nucleus response projecting to the brainstem. But the The effects of avoidance on creating resilience engage the ventral medial prefrontal cortex and are dependent on plasticity-related changes within the ventral medial prefrontal cortex. So in a way, we're augmenting this ventral medial prefrontal cortex inhibitory circuitry to bias you towards having a reaction and some resilience down the road versus a classic conditioned threat reaction. And consistent with this in humans, we've been able to show enhanced bold signal in the ventral medial prefrontal cortex and striatum during avoidance versus extinction. And this was work by Kate Hartley and also Emily Bucca. So the last technique that I'm going to talk to you about today to try to enhance the persistent control of threats that are no longer adaptive is what we call novelty-facilitated extinction. And just, you know, to promote sort of complete openness, we discovered this by accident. But now we're trying to figure out why it works. But let me explain to you what it is first. So part of the reason why we think it works is one of the things about extinction. Extinction is really a weak form of learning relative to acquiring a threat reaction. It results in a lot of uncertainty. Here's this thing that used to hurt you or potentially hurt you, and now nothing's going on. What's happening? So we thought that perhaps by maybe reducing the uncertainty in a way, by substituting the unconditioned response, the unconditioned stimulus with a neutral stimulus, we might be able to reduce recovery of threat reactions. And I should say, this looks a little bit like counter-conditioning. Counter-conditioning is now where, you know, it used to be a shock and now it's a reward. We know that counter-conditioning results in robust recovery of threat reactions. This is really something that's neutral, that's sort of meaningless. And just to provide some support for the notion that uncertainty might be linked to threat recovery, here I've plotted a measure of spontaneous recovery for a conditioned response and a measure of reinstatement for a conditioned response in humans. And on this scale here, I've measured intolerance of uncertainty. So people that have a higher intolerance of uncertainty show greater spontaneous recovery and greater reinstatement. Intolerance of uncertainty is really a trait-like measure that looks at how much you can deal with uncertainty in the world. So the way this paradigm works, participants come into the laboratory. One stimulus is paired with shock, another one never paired with shock. Half of the participants then undergo standard extinction training. They see all the stimuli, no shocks. The other half of the participants undergo what we call novelty-facilitated extinction. So now, every time they would have gotten a shock, instead they hear a neutral tone. And then we do a test of recovery on the second day, and there's no tones presented on the second day for both groups. So here I'm showing you the end of acquisition for both groups. This is, again, the conditioned response, the CS plus minus the CS minus. We see robust acquisition in both groups. Here's the end of extinction. We see robust extinction. We only see evidence for recovery, significant recovery, in the group that underwent standard extinction, not the group that underwent novelty-facilitated extinction. And this is one of the very few studies where I could say, hey, Joe, you know how we've been taking your paradigms and doing them in humans? How about you do this one in rats? So we did it in rodents too. I'm not going to walk you through it, but except to say that we were able to replicate this in rodents as well. So if you if you give a tone instead of the foot shock during extinction, we see less evidence of recovery, freezing response in this case in rodents. And this was work by Joey Dunsmore in my lab and also Vin Capizzi, who was a postdoc with Joe. So just to sum up this second part of my talk, I've suggested to you that extinction and cognitive emotion regulation are effective, clinically relevant techniques to control maladaptive threat responses. But the problem with these techniques is that threat is susceptible to relapse following stress, the passage of time, or changes in context. The targeting reconsolidation may prevent this relapse by reducing the dependence on prefrontal inhibitory mechanisms, but success with this technique is subject to boundary conditions, and clinical translation may be difficult. One boundary condition may be that these techniques are not so successful with generalized threats. That stressor controllability can enhance persistent threat control by augmenting prefrontal inhibitory mechanisms, and that novelty-facilitated extinction can enhance persistent threat control, but the mechanisms are still unknown. So we're actually trying to investigate this more now. One of the things we think might be happening is essentially that the tone may actually strengthen extinction learning, perhaps by inducing some type of surprise, so increasing associability, for those of you who know these sort of learning theory type things. So just to give you the broad overview, I hope I've convinced you today that animal models of threat conditioning provide a good basis for understanding human fear learning, but I think we always have to be mindful of the fact that the complex human social environment and the symbolic means of communication that we've developed has been adapted for us. It's led to greater flexibility of threat learning systems in humans than other species, and this can be both a blessing and a curse. And that this flexibility of human threat learning, our ability to imagine threats, may provide unique challenges in translating research from animal models to the treatment of clinical disorders. So I need to thank the people that paid for this work, the people that did and inspired this work, and all of you for your attention. Thank you.
So we have a couple of minutes for questions, and we'll start with Ed here.
So Joe Ledoux has been arguing recently that some of these amygdala implemented emotional SCR-type responses may not really reflect the full aspect of the emotion. For example, the subjective feelings and so on.
Yes.
And so I'd be very interested to know, what happens in your studies to the broader aspects of emotion.
So, Joe and I talk about this quite a bit, as you might imagine. He kind of gives me a pass because I study humans and I can actually measure subjective feelings. And granted, in most of these studies, we did not measure subjective feelings. When we did, for instance, the study with looking at stress and emotion regulation, we found a correspondence between skin conductance and subjective threat responses. But I completely agree with Joe for a couple of reasons. One, I think Joe's trying to be very precise about what it is that he's measuring. And two, we do see dissociations between threat reactions and subjective feelings. And one of the, in the recent study by Rothbaum et al., in which they tried to target reconsolidation in phobic patients using our technique of a reminder and then exposure, they did find some changes in skin conductance and heart rate in the reminder exposure group, but that didn't extend to clinical symptoms, right? So I understand where Joe's coming from with this, and I do think it's important that we start to look at both a little bit more.
Great talk. Do you have a sense of whether when targeted reconsolidation is inefficient, it results in greater generalization of the fear memory over time?
Oh, so in other words, when it doesn't work, do you get more generalization?
Yeah.
I actually don't know if that's the case. Marine in the back has an idea about this. Marine has something to say about that. Yeah, Marine, what do you want to say?
Where are you?
Because he did that one of those studies. So, he's going to talk about episodic memory, which is something I didn't talk about, but he's been measuring that.
Yeah. So actually, in the reminder extinction study that Liz already showed, we don't see a prevention of the recovery of threat responses, but you actually see an enhancement of episodic memory for events that happened before. Yep. So you get an enhancement of a generalization of memory for in a different domain of memory, really. So you can see that reconsolidation actually strengthens memory. We found that in some other studies before as well.
So it strengthens the sort of hippocampal component of the memory in that case, which is actually something that Christina Alberini, my colleague in the back, has been talking about a lot, as reconsolidation as a mechanism for actually strengthening memories.
Other questions?
One right here.
Oh.
Thank you. It's great to see how you're extending this into the clinical world recently. We're
trying to.
Novelty.
I can't say we've done so successfully yet, but we'll get there. So.
Well, you get an A for trying.
Yeah.
So the novelty-facilitated extinction, it kind of is similar to reversal learning in the sense that you have a CS plus instead of being followed by now transitioning to the CS minus, reversing the stimulus contingencies in reversal learning. It's more that you have a CS plus followed by a new stimulus.
Yes.
So is there anything to learn from the reversal learning literature that would help inform mechanism on why this novelty is working?
I mean, not that off the top of my head, not that I can think of. I mean, we've done some reversal studies, but here, in reversal, it's sort of like an extra challenging inhibitory, task, right? Because now this thing that was safe is now not. So it's actually, you got to learn new things, right, about this. So I'm not so sure I'm thinking about it in that regard. What we are thinking about in this is that if we look at learning models, extinction learning, one of the things that we find is that, as something becomes more predictable, you don't update your prediction error as much. So we think that we're sort of maybe changing some of the predictability of what's going on, and that's actually enhancing your ability to learn the safety signal. We don't really know if that's the case, but that's kind of our working hypothesis.
Okay, I think we should thank Dr. Phelps one more time.